All right, let's get started, guys. So chapter 10, we're finally here to the metabolism chapter. So much of this information applies outside of this class. It's like not even funny. So, but yeah, it's one of my favorite topics, and I hope that you guys, find it interesting too. I think it's incredibly fascinating. So let's get into it. Let's get into the boring part first. First of all, metabolism in general, that's all of the chemical reactions and the physical workings of the cell. So yeah, when people are talking about your metabolism being slower than somebody else's or whatever, that still applies. They're still talking about chemical reactions, but it's actually a bigger picture than that, okay? That can be divided up into anabolism and catabolism. And yes, like anabolism, I'm literally referring to anabolic, like anabolic steroids, okay? Anabolic steroids, what do you do with those? You take them to build muscle, right? So you're building something, you're creating something. In anabolism, you're building something. It's biosynthesis. You're creating bonds or structures, new muscle tissue. All of that is an anabolic reaction. You're building things, so you need energy to do that, right? Just like those bouncy balls. If you want to pick them back up and put them back in their container, that needs energy. So that's sort of like building something too. Next, we have catabolism. Catabolism is breaking bonds, and usually the body's going to be doing that in order to harness the energy involved in what was those bonds, okay? The way I remember the difference, if you can't remember about anabolic steroids being building muscle, then at least hopefully when you see catabolism, you'll think cat, and that cats are going to destroy everything. So they're going to rip everything with their claws and tear it apart. That's catabolism, all right? So we're breaking bonds to release energy. That's what the cats are doing, man. All right, that's the whole point. And then when I say releasing energy, we're going to collect that in the form of ATP. Today, the whole point of what we're talking about is how that even works, okay? All right, anytime you have a reaction and you want it sped up, and there's something there that will help speed it up, those things that help speed it up are called catalysts. Catalysts help overcome the activation energy. The activation energy is just the energy that has to be put in to make that reaction go. Essentially going to need more energy if you don't have the catalyst there to help, right? So there's other ways you can increase or help overcome activation energy. Heating things up. We kind of already know that, as far as doing experiments in chemistry and everything like that. So that makes sense. Heating something up excites the molecules. They're going to be more active. Cool. Increasing the concentration of the reactants to increase the rate of molecular collisions. Literally on the chemical level, the more stuff you got in there going on, the more likely it is to have the event occurring. And then finally, another way to overcome that energy is just like what we said, using the catalyst. So those are ways you can do it. I really want you guys to focus more on the catalyst aspect of it, because organic molecules, typically proteins, that help overcome activation energy as catalysts are known as enzymes, okay? So for all intents and purposes, I know it says on the board, but for all intents and purposes, for you guys, we're just going to look at it that way. Enzymes are always going to be proteins, okay? And anytime you see a molecule that ends in ACE, DNA ligase, DNA polymerase, RNA polymerase, all of that, ACE, 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 ACE. Those are all enzymes if you hear ACE at the end, okay? So if I were to put, on the test, what type of biological macromolecule is this, and ended in ACE, you should be able to choose protein just automatically because it's an enzyme. All enzymes are proteins, all right? So enzymes are catalysts in our biological, biochemical situation. They have an active site. That's where the molecule they're going to be acting on will bind. There's no big old surprise there. If you have an enzyme that looks like this, I'm hard to know about it drawing this stuff, but the enzyme that looks like this, and this is the active site. And then here comes the thing that binds in it that we're going to be changing, right? So what will we call that? That's the substrate, okay? All right, so we're getting into enzymes. Substrates, they bind to the active side of the enzyme. That enzyme helps overcome the activation energy for whatever the chemical reaction is that we're doing with the substrate, okay? You might be breaking it apart and creating two parts out of it. I don't know what you're doing with it, but that's a possibility. That's one example. But in either way, there's the active site, and that's where all of this is going down. These guys are proteins, and their structure is pretty much just as important as the chemical aspect, like their chemical makeup, okay? So their structure determines more what their function is. So they're highly sensitive to pH and temp changes, usually high temp specifically, okay? Why is high temp important? Why specifically high temp and pH? Because we're talking about denaturation. We mentioned this when we talked about it in chapter 8, right? Denaturation of the DNA, heating them up in the PCR. Heat up the DNA in the PCR machine and it splits the strands apart. That's denaturation of the DNA. What bonds were holding those two strands together? Hydrogen bonds, weak bonds. The important part of what makes a protein, if it's not the very few covalent bonds holding it together, is mostly hydrogen bonds determining that, okay? That's true with a lot of interactions in the body. So it's hydrogen bonds that are determining the shape and determining this shape in there, even specifically, that's going to be very sensitive to temperature, heat. That's going to denature that, right? It's affecting those hydrogen bonds. pH is also affected by the hydrogen bonds, or it's going to affect the hydrogen bonds because you're having a high increase in hydrogens in the area or a massive decrease in hydrogens in the area or increase of hydroxyl ions even. So those things can cause basically a complete downfall, change the structure of these enzymes so that they can't distort their shapes, so they can't do their normal functions. That's really important. They can be regulated in a lot of different ways as well that we're going to talk about. So the substrates, that's what the enzyme is acting on. The enzyme itself, while it's doing this, doesn't become part of the products. So it's not going to be, we don't consider it in the chemical reaction necessarily, okay? It's not part of the reactants, not part of the products. It's just there to help it go. It's not going to be used up by the reaction as a result of that. It's not getting involved in the actual chemical reaction. And therefore, it can function over and over and over again, okay? That is excellent because that means less expenditure for the cell, just being able to reuse as opposed to make more protein in order to continue with something. It's excellent. We have two kinds of enzymes, simple enzymes and conjugated enzymes. All right, so simple enzymes is just the protein. Now maybe you're wondering what that next one is going to be. Conjugated enzymes. We have the protein and some other crap helping it, okay? All right. That gets us to those conjugated enzymes get their own little terms. We're going to break them down. The whole thing that functions, thankfully, it's called the whole enzyme. You can think of a holodeck if you're thinking of like, you probably ever watched, the Star Trek or something like that with Enterprise and all of this with the holodeck. But that's what I think of when I see this. What the thing that it also sounds like is whole, right? So it's the whole thing. So think of it that way. Holo enzymes, the whole thing, the one that's going to do the work, okay? You need to be whole in order to do the work. First part, the apoenzyme. The apoenzyme is the protein portion, okay? The cofactor is the non-protein portion. So what can it be? It could be an organic molecule of some kind. If it is organic, then it is a coenzyme. A lot of times this can refer to vitamins or other organic molecules bound with the vitamins, okay? Usually if you need a vitamin in your diet, it's for enzymes to work, okay? They're cofactors for enzymes. Then we have our inorganic, which usually is going to be our metallic ions. So we're talking about a protein that requires maybe, for example, iron, let's say, in order to perform its job. Without that iron, it can't perform its job. That sounds an awful lot like hemoglobin to me, and it should to you as well. That's exactly what we're talking about here. That protein cannot perform its job without the iron there. How are you going to carry around the oxygen and stuff? Yeah. So that's the idea here as well with these enzymes. So we have different kind of, different kind of situations going on here where you might need the metallic cofactor. You might need the organic cofactor, which we call a coenzyme, or you might need both in order to function. It depends, okay? Usually, whenever we have our cofactor binding onto it, wherever it binds, so your metallic ion, let's say, that will cause it to change its shape so that now it can interact with the thing. It's like if, I don't even know, it's gonna get weird really fast, but let's do it anyways. It's like if I was sitting in a chair, let's get a chair. Let's not make it weird, pretend like we're sitting. It's like if I was sitting in a chair, and what I want to do is hold my fidget spinner. That's what I'm made to do. I'm sitting here in my chair and I can't pick it up because my hands are stuck on my knees. Okay. You put my nice fancy necklace on me. That's my cofactor. Now I can reach over and grab this. That's sort of what this is about. Okay. So we've changed the shape or allowed it to move somehow by interacting with that apo enzyme. Okay. All right, so obviously we've talked about proteins before, and I know that you love hearing about them, but we have the different levels of the proteins. Now they're fine. That crap is finally coming into play somewhere, all right? We have our actual primary structure. That is just only the amino acid sequence, okay? That's all that is. Remember in DNA, how we had A, T, C, G, blah, 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 blah. That's just the sequence of your nucleotides, right? With proteins, our sequence is going to be your amino acids, proline, glutamine. I can't even think of them right now. Phenylalanine, whatever they are, right? And whatever that is, amino acid, amino acid, amino acid, amino acid, that's your primary. Your secondary is how those amino acids are kind of interacting with each other, and they'll create these structures that are kind of predictable. The alpha helix, or the beta sheet. And then our tertiary, the whole thing starts interacting with the beta sheets with other beta sheets, or beta sheets with rando parts, or whatever it is. Create some covalent bonds by creating disulfide bonds, whatever. Once that is fully folded, that polypeptide, that one polypeptide is fully folded, that's the tertiary structure, okay? You might think, well, that's when it's done. Absolutely not, man, then you're not understanding. That is with the one polypeptide, y'all. An enzyme isn't made of one polypeptide. Don't think that. The quaternary structure is when all of the associated proper polypeptides have come together and nestled in together and interact together to form the functioning enzyme. Okay? That's the quaternary structure when that happens. Now, maybe that quaternary structure needs iron in order to function. Well, then that would be the apo enzyme. The iron would be the cofactor. And the whole thing is a holoenzyme. And that's what actually does the work. So it's just to bring it all home. All right. Here's a picture as if this is helpful at this point. We have the enzyme, really the purple enzyme. We have the thing that fits and the thing that looks similar but doesn't quite fit. It can't. So this fits and it gets its little products made as a result. So again, it's like if I have my cofactor, my little necklace is on, I'm able to grab my fidget spinner, okay? That's step number one. When I grab the fidget spinner, the enzyme, when it grabs the substrate, causes it to change shape. So like if I stand up when I grab the fidget spinner, okay? And when that happens, that allows it to do whatever it's doing. Break the bonds, or maybe even make bonds, depending on the enzyme, okay? So it's all depending on those chemical interactions. And when I say it's standing up, it's moving, it's doing these things, those all governed by chemical interactions, usually hydrogen bonds interacting with one another. Yeah, that's how important that crap is. So our metallic cofactors, iron, copper, magnesium, manganese, zinc, cobalt, selenium. These are our trace elements in our diets. You need them. Sometimes they will even be participating in the chemical reactions. Usually they're going to bring the active site and the substrate closer together so we can perform our job better. And then we have it says activate the enzymes. Like I said, if I'm sitting there and my, I don't know, necklace, my iron comes and binds with me, now I can grab my fidget spinner. I couldn't have done that without that help. I don't know, not in reality, obviously that makes no sense, but as far as the enzymes go, all right, let's go on to coenzymes. Remember, coenzymes are just a type of cofactor. Cofactors, we have two types. We have the inorganic, like those metallic cofactors, and then we have these coenzymes, which are organic. So it's just a type of cofactor, right? They're gonna do a similar thing. I don't know what else to tell you. They do a similar function, but they're organic molecules now. A lot of times, vitamins. Cool. All right, so we understand a little bit about the structure of an enzyme, what it might need in order to function, and everything like that, how important the shape is, and how sensitive it might be to change in its hydrogen environment. Here are these six main groups of enzymes, okay? Two of them I will not ask you about. Now, I have not written the test for this exam yet. We're in the process of it. I've got some of the questions, about half of the questions. Now I'm trying to, I don't want to say spice it up. That's not right, but yeah. Give a little bit of a flair to it, I guess, without being too complicated. But I'm not going to ask you about a lyase, but they do have to do with double bonds. Okay, whatever. And I'm not going to ask about isomerases. These are enzymes that are going to change the shape of a molecule. It's still the same molecule, but it's a different shape of it. Like, you know, L and D versions of certain molecules. I'm sure you've heard of those in some of the drugs you take, but I am not going to ask you about those. So the ones I might at least ask you about, the ones that are important, oxidoreductases. Oxidoreductases. Maybe you're looking at this and you're thinking, this looks an awful lot like redox. If you had chemistry, your heart is probably sinking right now because you're like, redox reactions. Yeah, we're gonna do that, y'all. That's why you have to have chemistry before you come to this class. We're gonna do that. The absolute most important aspect of aerobic respiration relies on redox. It's all driven by electrons. And if you don't understand about that aspect of it, you don't appreciate that, then you're never gonna get what we're gonna explain to you. All right, so if I were to tell you, because you definitely understand about redox reactions, dehydrogenases, these enzymes, they are a type of oxidoreductase. Why? Because hydrogens typically in a biological setting are going to be associated with an electron somehow, either leaving one behind or taking it with them. So that's why dehydrogenases fall under that, okay? Transferases, we're going to put one group onto another group. Somewhere in, I believe it is the Krebs cycle, we are going to be moving a phosphate group from some crap onto ADP to turn it into ATP. That is not the main way that we make ATP, to be clear, to be very clear, okay? But you can make it that way. But we're just moving a group from one place to another, yeah? That is what transferases do. Hydrolases, we are now cleaving or breaking bonds. I just can never see, it's like this too little. And I need to be wearing reading glasses anyways, so I don't have them. Hydrolases, they're gonna cleave bonds on the molecules with the addition of water. That's where the hydro part comes from, okay? It's water part. So we're gonna be adding water into this reaction in order to cleave some bonds. That's all this is, okay? And then the next one, wow, we skipped over to ligases. These guys catalyze formation of the bonds. We have to add ATP, right? Anytime you hear me say we're making a bond or building something, you need to be thinking that needs energy. Where are you getting it from? Okay. So we're getting it from ATP. And as a result of this whole reaction, we will have removal of water. So we have water as a product of these reactions. Those are ligases. That should sound familiar. Yeah. DNA ligase, one of the most important enzymes that we've talked about so far. Helps seal together your Okazaki fragments, all that stuff. Okay, you make your little cuts with your restriction endonucleases, and you slap your things together. You need to seal it up with DNA ligase. So you can see quite clearly hydrolases and ligases are kind of, I want to say opposites, but yeah, right? All right. All right, what are we doing with our oxidoreductases? And this is kind of a bad place for this slide, but it's here, so we're going to mention it just briefly. Our oxidoreductase, these are going to be our redox. It's not really how you do it, but okay, it should make that small and that big, but whatever. Redox reactions. Okay. So these are redox reactions, at least the ones that are important as far as what we're going to be talking about. Okay. So oxidation, loss of electrons, or loss of electrons. Yes. Okay. No. Okay, that's not actual erasers. Let me do this. I'm adding the O in there. Loss of electrons is oxidized, or gain of electrons is reduced. So what am I trying to spell here? I say Leo, the lion, by the way. Leo says GER. That's how I remembered it. That's how I learned it. When I was in AP Chem in high school, this is how I was taught this, and that's how I remember it now. You do what you want to do. There's another option that you have here. Oil and rig. Oxidation is loss. Reduction is gain. If you think I'm just saying this, okay, cool, that's nice. That's so you know what oxidation and reduction are. You better believe I'm going to ask you on the test which molecule was reduced and which molecule was oxidized. I am going to ask you that on the test. Be prepared. Okay? Because if you can't understand about oxidation and reduction, I promise you can't understand about aerobic respiration. Okay? So be ready for it. This is how I remember it. If you need to write it down on scratch paper, good on you. I have to do that also sometimes. If there's like a equation there and somebody's like, okay, which one was reduced? I have to be like, okay, what is reduced? Oh, that's gaining electrons. Sometimes I can't recall that, so I'll write down Leo says GER so that it says it for me. That way I don't have to recall it. That's going to say it for me. Do what you want to do, but oxidation is the loss of electrons. Anytime you are giving up an electron or having it taken away from you, you are being oxidized, all right? Reduction, gaining electrons. Anytime that you are stealing away an electron, right, or being given an electron, that's you're being reduced. What are some very obvious things we might think about in this situation? We might think about sodium chloride. Oh yeah. So remember, that is an ionic bond. Remember how one of them donates an electron to the other? Uh-oh. That's a redox reaction, right? So essentially, to create that ionic bond, we have a redox situation going on. So sodium donates its electron and becomes positively charged. We know this. And that's why chlorine is negatively charged. It takes the electron on, right? So this one lost the electron, so therefore it is oxidized. This one gained an electron, therefore it is reduced. It really is that simple. Don't overcomplicate it, okay? It's just whether you're giving up the electron or you're gaining the electron. Now, maybe perhaps you're sitting there and thinking about, yes, but what if we're talking about reducing agents and oxidizing agents? I don't know if you guys know what I'm talking about right here, but that gets very complicated very quickly if I'm asking you to tell me about reducing agents versus something that was oxidized and stuff like that. I'm never going to ask you about that because that's ridiculous. It might be something useful. It is useful knowledge-wise, but it's just too much. So we're leaving it, okay? So we're just only worrying about what is being oxidized and what is being reduced, because I am going to be saying it over and over and over again, okay? We have these molecules, NAD and FAD. These guys are going to be carrying around electrons for us later on whenever we're talking about the electron transport chain and everything. These guys are coenzyme carriers. So what do we know about them? We know they're organic molecules, right? And there are enzymes that interact with them that require them to interact, basically. But the important thing that I want you to know about these guys, NAD and FAD, they're going to carry electrons around for us. That's all really you need to know. Am I even going to ask you about what NAD or FAD stand for? I don't care, okay? I don't care. It's nicotinamide, in case you're wondering, but I don't care. So you don't need to know that. I just want you to understand the process. I feel like that's the most important part. So some enzymes to understand about, exoenzymes and endoenzymes. We learned a little bit about the exoenzymes with the saprobes. If they want to eat something dead, they'll exude these exoenzymes to break down whatever's around them, and then they can absorb up the nutrients, right? So that's an exoenzyme. It's outside of the cell. We also have endoenzymes. Almost all of your metabolic pathways are going to be driven by enzymes inside of the cell. So endoenzymes. We can regulate enzymes. They can be constitutive, which means always on. They're just always around, same amount, just always regularly. It's always there. Not more, not less. It's just there. Then we have the regulated enzymes. These can be turned on or off. And now here I am talking about the enzymes themselves. Can we also control this at the genetic level? Absolutely. But here we're talking about the enzymes themselves. So here we have a constitutive example. We just, however many purples are there, is however many purples are there. You can add as much substrate as you want. You ain't getting any change. That's just how many is there. All right, regulated enzymes. We have two different kinds. We can induce the enzyme. If we are inducing it, that means you get more substrate added in than usual. We're going to make more enzyme in response. If we are repressing our enzyme, it means we could have all this ton of enzyme. It's all interacting with our substrate, but maybe now we've done all of our work on the substrate. There's no more left to work on. We don't need the enzyme anymore. So it goes away. That's all. Okay. It's just reacting to its environment. Those are the two possibilities. I know we have talked about operons, which I'm sure you guys are just intentionally blocking out of your memory. I completely understand that because I hate an operon. But operons is whenever we have those sets of genes in bacteria, it's only in bacteria, that can be induced or repressed, just like this. But in our cells, we can do that too with our enzymes, but we just don't have them in a whole set of genes. It's just like one gene. Okay, so it does still happen. It's just not an operon. Remember, bacteria can have gene after gene after gene all linked together, controlled at one point. That's an operon, all right? Sometimes we have enzymes that are involved in disease. Maybe they cause tissue damage. Maybe they do something bad. I don't know. But if they are associated with making disease happen, pathogenicity, then those are virulence factors. So virulence factor is anything that can be causing pathogenicity, including enzymes. So I don't want you to think that all virulence factors are always enzymes. That's not true, all right? Anything. We even said about how flagella, right? We learned about that in lab. Flagella can be virulence factor. That's not an enzyme, obviously. But these enzymes, they function as toxins. They cause damage. That is pathogenicity. We have streptokinase, streptolysin, elastase. I'm going to focus on two of them because I feel like they make the most sense to everybody. Collagenase, lipase, and penicillinase. All right, so obviously the circled ones I'm going to talk about right now. We all know, we've all heard at least of collagen, right? Involved in, keeping you from getting wrinkles, keeping you from getting cellulite, and whatever else. That's what collagen we think it's for. Not actually what it's for. What it's really for is your extracellular matrix and maintaining tissues, right? It does decrease over time as you age. So that support of your tissues decreases, and then you get cellulite and wrinkles and stuff. So that's what collagen is really for. So now I'm telling you this enzyme, this toxin, is called collagenase. So we know that's an enzyme because it ends in ase. So it's protein too. So what do you think it's probably doing? Breaking down collagen, right? So collagenase breaks down collagen. That's part of your extracellular matrix, what holds your cells together. That can make it hard for microbes to get into your body, deeper into your body. So it breaks it down so it can get in deeper into your body faster, quicker, easier. That's what collagenase is, okay? It's a terrifying thing to think about, actually. A lot of bacteria have this, including strep. It does this quite well, okay? All right, the second one, now that I've mentioned that, I'm sure this second one is pretty clear to everybody. Penicillinase, what do you think it does? Breaks down penicillin, right? So here, this penicillinase, this is a drug resistance gene, right? So that makes you resistant to penicillin. You're breaking it down. You can't kill, my cell wall. Penicillin, by the way, is associated with preventing cell wall. formation in bacteria. So they're just going to say, no, 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 I'm going to go ahead and keep that cell wall. Let me go ahead and break you down, son. So it does that quite well too. So that is a drug resistance gene. Remember, a lot of drug resistance genes are going to be carried on plasmids. Remember how plasmids, circular little bits of DNA that are not required by the cell, can be helpful with their DNA. Okay, they can be transferred from bacteria to bacteria via horizontal, transfer. Yeah, I could go on and on about that, but we're going to move on. Denaturation, we've already brought it up. So our weak bonds, our hydrogen bonds, that's shaping our apo enzyme, the protein part of our enzyme, those are going to be affected by heat, low or high pH, and also some chemicals like phenol. Phenol is very well known for disrupting protein shape. Okay, so our shape gets ruined. That ruins the activation site, so we can no longer bind at that active site. So we can't do our work anymore. And that can even lead to cell death if it affects enough of those proteins. So the same thing as if I just literally shove you into an oven and say bye, even if that oven doesn't create flames themselves, which is what we think makes something hot, it doesn't have to. If it gets hot enough to denature your proteins, then you'll die from that. That's it. Even if I give you all the oxygen and the food and the water in the world, your proteins will denature and you will die from that. That's how important this can be. Let's see. We're going to talk about the different kinds of metabolic pathways. Go figure that they could be all sorts of different complicated kinds. Linear, just straight A to B to C to D, whatever. One thing relies that the other one gets made in order to go on, that sort of stuff. Then we have cyclic, where something that we're starting with is probably going to get made again in the end. And that's how that would work. And then we have the branched ones, divergent and convergent. So we know what it is to converge or diverge. Yeah. If you're diverging, you have this molecule splitting into these. We've broken bonds. If we break a bond, you can get energy out of that. You break the bond, that's catabolism. All right, convergent, we're converging. C plus Z equals M. That's our reaction right there at that point. So we are making bonds there. We are creating something. That could be amino acid synthesis. It could even be putting amino acids to one another. No matter what, if you're building a bond somewhere, you need energy for it, right? Right. Let's talk about how you will control enzymes other than talking about increasing or decreasing the amounts that you have. Let's talk about controlling them directly. Some molecules can mimic the substrate, right? Can mimic this. So that might be our substrate. Let's just say, make it easy on us. And this is the thing that is going to mimic our substrate, all right? We will call this an inhibitor. And obviously this is going to just pop in right here, right? It does not interact the same way as the substrate. It's not going in there to be cleaved into the products or whatever. It's not there for the normal stuff. It's literally there just to sit there. It's like it's spiteful. It's going to be there, be like, I hate you, substrate. I'm just going to go ahead and sit in your spot so you can't use it. Basically what's going on, right? They will sit there for a very long time, actually, so that this enzyme can't do its job, right? It can't ever cleave this. So it's kind of screwed at that point. That is competitive inhibition. That's because our inhibitor is competing at the active site, okay, with that substrate. We're competing at the actual active site with the actual substrate for that position. Next, we have non-competitive inhibition. Lucky you, as far as inhibition goes, there's only the two types, competitive and non-competitive. Competitive, we already know we're competing for the active site. So guess what? Non-competitive, we're not competing for the active site, all right? In this case, instead of having a mimic that binds at the active site, maybe your substrate comes in here, whatever, and we bind here, and now we're going to get cleaved into two parts. Tiny triangle, and then weird round part. I don't know, these two parts, let's just say. That's what they look like, cool. And eventually, so the cell uses these up for whatever they're doing. Making amino acids, or building parts of the cell, or whatever. But now the cell does not need these anymore. So typically, what's going to happen is that product, as it builds up, Now it will, since it's not being used by the cell, it will come over here and bind at this other place. So it's non-competitive. We're still going over there and basically binding with the protein, the apo enzyme, but at a different place. Okay. When that happens, that apo enzyme, who normally is going to be reaching for and acting with all these fidget spinners or whatever, something comes along and let's say binds at the back of the knee for me, I don't know, but it causes me to sit down again. So remember, if I'm sitting down now without the other help of this, then I can't get the fidget spinner. That's the idea. So it's changing the shape of the actual protein so that it can't interact with the substrate anymore. Not binding at the active site, but it is interacting with the protein enough to cause it to be unable to interact with that substrate. Yeah? So you don't need the products anymore. That's usually the feedback mechanism that we'd be looking for. Here's some pictures of what I'm trying to explain. Did I leave? Yes, I did. You count on me to leave things in the weirdest places. All right, normal substrate is this brownish color. I don't know what it's called. So this will be competitive and this will be non-competitive. We'll get to both of them. In the competitive one, we have our enzyme. Normally we bind in here in our active site. Substrate gets cleaved, we make these products. Cool. However, in competitive, we are going to have that pink, light pink, similar shape, binds in the same spot, makes it so the enzyme can't do its job normally with the substrate, sits there on it, so it can't. And now we can't get our products made. These greens don't get made. Okay, that is competitive. Non-competitive, we have our enzyme over here. It has our substrate same color. Normal situation. Normal situation, it binds and we make our product. In this case, yellow nose shape. I don't know why I typed that way up, but it is. Nose shape. Anyways, you get enough yellow nose shape, it will bind somewhere else at the other spot. The regulatory site, you could call it that, or you can call it the allosteric site. Those are both accurate, okay? But they're not the active site. Anyways, it causes a change in the shape. See here now how our substrate can't bind. And that's how that will work. Enzyme repression, this is something else now. We talked about competitive, we talked about non-competitive. Now we're at the genetic level. That's what this is going to be, okay? Somehow, some product you're making can interact with the genes somehow. So here we have our DNA being transcribed into RNA, RNA translated into the protein product. We make our protein, fold it into the right enzyme structure. It's going to interact with its substrate and make its products. You build up enough products, they're not being used by the cell anymore. They will come over here and interact with the DNA at a certain spot and shut down the synthesis of that product. So it's literally at the actual genetic level. That's all this is. But a similar concept. All right, if you're going to induce, it's pretty much going to be essentially this reverse of what we were just talking about in literally this picture. It's going to be the reverse of that. So you can use it to repress or you can use it to induce. That's all this is saying. Cool. All right, energy in our cells. They can, anytime we're having any reactions in our cells, they're going to be exergonic or endergonic. And please tell me you recognize those words, right? Exergonic, energy is coming out. Exo, I'm going to release that energy and it will be available for work, right? Endergonic, endo, into, require the addition of the energy to move forward. So exergonic reactions where we're releasing, these are catabolic reactions, yeah. Endergonic, they need energy. These are our anabolic. It's just talking about the actual chemical reactions themselves at this point, okay? Let's get into those redox reactions again, all right? Anytime you have a redox reaction, the reason we call them redox reactions is because you always have both. You always have one thing that is reduced and one thing that is oxidized, always. The electron donor and the electron acceptor. That electron doesn't go into the ether. Never forget that. The electron has to go somewhere in the end, all right? That's very important when we get into aerobic respiration. So these oxidoreductases, those enzymes that are involved in this, they're usually going to be interacting with those coenzyme carriers, NAD and FAD, whatever. They're carrying our electrons. They're going to take those on and move to the next step. We've already talked about sodium giving its little electron to chlorine. Now it's positive, and now that's negative. So this lost, so it is oxidized, and this gained, so it's reduced, right? Just reprising that so we're aware again. Here's NAD. This is what you wanted, yes? NAD, whatever. It's our little molecule. If it interacts with a hydrogen, we're talking about an actual covalent bond whenever we look at it this way, okay? It becomes NADH. So there's an actual covalent bond there. You guys remember what covalent bonds are? We are sharing electrons, right? So we're sharing electrons. There has to be electrons there. So that H brings an electron in. That's what that hydrogen is doing here. This other hydrogen that's involved in this whole mess is a proton. It doesn't have any electrons, right? That's important to remember as well. I'm never going to have you worry about this shape. All right, so moving on. We're going to use the movement of electrons from one place to another to create energy. We're going to harness that. So I'm trying to see what I care about here. We already said this, NAD and FAD going to be carrying around our electrons. That's in all aerobic respiration, okay? And anaerobic. Plants, they have another guy, NADP, in addition to these. That'll also carry, but it's only in photosynthesis. Right, let's go on to our catabolic pathways, where we're going to get our energy from, right? The most important thing that you guys are going to worry about, aerobic metabolism. If you have not taken physiology yet, if you are currently in physiology, I don't believe you guys have gotten to metabolism yet. You will, okay? You will learn a little bit about this stuff. I love it, so I don't mind it. Okay, so aerobic versus anaerobic, right? We already learned these terms. Aerobic, we know that's going to have oxygen involved. Cool. Anaerobic, not oxygen, right? That's really all you need to remember about these terms here as well. Anaerobic, no oxygen. All right, something else besides oxygen. That's all you need to worry about with that. So it says here that oxygen is our final electron acceptor. So I told you electrons have to go somewhere. That's where it's going to go. Okay, we'll get to it. Don't worry. Reminding you about adenosine triphosphate, ATP. It's a 3-part molecule. Adenine, that's the base, the nitrogenous base. Ribose, 5-carbon sugar. It's ribose, not deoxyribose. Okay, and then 3 phosphate groups. Very important because these phosphates hate each other. They all have the same, like they're all like north ends of a magnet. They're constantly pushing each other apart. So that third one is really wanting to leave. And so if we release that third one, we can use the energy involved in that bond breaking to fuel other reactions in the cell. So that's what the importance is of ATP. I'll tell you just how important it is here in a moment. So ATP is the primary energy. We can make it via substrate level phosphorylation. It's a fancy way of transferring the phosphate group from one thing to another. Okay, that's substrate level phosphorylation. You can also make it via oxidative phosphorylation or photophosphorylation. So there's only 3 ways you can make ATP for the cell. Okay, the main one, the main biggie, oxidative phosphorylation. All right, that's what we're going to be talking about in a moment. Obviously, photophosphorylation, we're using energy from the sun to do a similar thing. Okay, let's get into these. We have aerobic and anaerobic. And remember, we have our fermentation organisms. Those are our facultative anaerobes, mostly. Remember, they can switch between. So if there's oxygen, they're going to be aerobic. If there's not, they ferment. It's a big difference with fermentation, okay? Massive, massive difference with fermentation compared to all of these other ones. Completely, massively different, okay? But they all start out with glycolysis. So glycolysis, all of them. All right. You guys ready to do this? You better read because I love it. All right. This is the setup of these metabolic pathways. Here we have first glycolysis. Notice how they all do it. Whether you're aerobic, anaerobic, or fermenting, all of them have glycolysis. Okay. There's production of ATP there. That's nice. Yields about 2 ATPs. So we're talking about fermenters literally only making 2 ATP from one glucose molecule. Now that is nuts. And I'll tell you why. because with aerobic and with anaerobic, you're going to take your little broken up product, shove it through the Krebs cycle, make a bunch of electrons, shove those electrons down the electron transport chain, and then make ATP. Here, we're going to use oxygen as our final acceptor, and in the other one, anaerobic, anything else, okay, to make ATP. Look at this, 36 to 38 ATPs, and then 2. Now, you might be saying, like, yeah, but how do you compare those? Let me tell you how. Those are each from one glucose molecule, equal. One glucose molecule. Push it through fermentation, you get 2 ATP. Same exact glucose molecule. Push it through aerobic respiration, you get 38. So way more efficient. So you can see why that is the preferred method. Now you can do something very similar in anaerobic, but just remember they're not using oxygen there as their final thing. All right, let's get into it. Aerobic respiration, we got a bunch of enzymes doing a bunch of work, changing up a bunch of molecules, breaking up a bunch of bonds, and harnessing that energy. That's really what's going on here. So let's get into that. Okay. This is glycolysis. Yay. It looks fun, doesn't it? It doesn't to me either. I hate it also, and I am a biochem major. So let's do this. All right, up top, we have glucose. The important thing that I want you to realize about glucose, it's got 6 carbons, okay? We're gonna go down, this is all just glycolysis, by the way. This isn't the whole of the thing. This is just glycolysis, okay? All right, the bottom down here, 3 carbon plus 3 carbon. So 2 times 3 carbon. We haven't lost any carbons at all. We've just shifted around some molecules basically to get to 3 carbon molecules. Yeah, literally the whole point of glycolysis. 6 carbon glucose molecule, breaking it into 2 3 carbon pyruvic acids, okay? That's the whole point. If you can't remember that and you want to know the rest of it, I don't know what to tell you. I'm not going to tell it to you because I hate it. So you don't need to know it. You just need to know. Glucose, 6 carbon, yields 2 3 carbon pyruvic acids. That's the whole point of glycolysis. Literally, that's the point, okay? For us. Now, if you're fermenting, the point is the ATP, sadly, but whatever. So up here, you can see we use an ATP and we use ATP. So we've used 2 already. And then we gain an ATP and we gain an ATP. And we do that twice per glucose. So now it's a net of 2. That's where that comes from. I don't need you to draw it. I don't need you to even point it out for me. I just want you to be aware of why we're talking about the 2, okay? That's where it comes from for those fermenters, because you have to know about those, because this is microbiology, it's not physiology, right? So I have to tell you about bacteria. Okay, what else do we do along the way? We make some ATP, cool. What else are we gonna do? We're gonna make NADH out of our NAD, whatever. Here's our electron carrier, NADH. Here's our electron carrier, NADH. So that's made here too, okay? NADH is made in glycolysis. It's not the point of glycolysis. The point is to make pyruvic acid, okay? So what's pyruvic acid for? What do we even give a crap of a pyruvic acid, right? If that's, I'm telling you it's the point, what the heck, what do you mean? What does that mean? Turns out pyruvic acid, which you get from glycolysis, which they've just summed up that whole chemical pathway in this one arrow here. But we've got our 2 3 carbon pyruvic acids. You can use these to make amino acids, sugars, and fats, stuff like that, if you want to. However, we're not talking about that here. We're talking about making energy. So what do you do with it here? You've got your 3 carbon crap here, pyruvic acid. You're gonna steal a carbon from it and other stuff, but you don't need to worry about it. And it's gonna become 2 carbon acetyl-CoA. And since there's 2 pyruvic acids, you'll get 2 of these too. So 2 carbon acetyl-CoA. I'll write it down here so you can see it better. Now, co means coenzyme, okay? Now, facultative anaerobes, the ones that are going to be doing fermentation, they're not going to get this far, all right? They stop at the pyruvic acid. But here we've got our 2 carbon acetyl-CoAs. Those are like the real goal, but this is an intermediate step. But it is important to recognize that it's there. Why? Where does that carbon go? All right. That carbon because we are going to make an NADH in that. While we're doing that, knocking off that first carbon, making it into 2 carbon instead of 3, we're going to release our first carbon dioxide molecule. Okay. So you've taken a 6 carbon sugar, glucose, broken it into 2 3 carbon pyruvic acids, and then knocked off these carbons with it to steal the electrons. And when you do that, the one that was left over, we're taking out as carbon dioxide. You see where that carbon comes from? That's what you breathe out. That's the only place where the carbon dioxide that you breathe out comes from. It doesn't come from anywhere else. It's only here. Okay? Now look at me and tell me how many times you breathe in a day. That is only for this. I'm not kidding. It's only for this. Okay? That's how important this is going to be. Let's move on. We know about NADH, whatever. In our little intermediate step, we kicked out a carbon, stole an electron, whatever. Now we're going to go into the Krebs cycle with our acetyl-CoA. Okay? Let's see what I care about here. We're just going to go straight into this. Okay, so our acetyl-CoA, just by the way, is right here. Here's our 2 carbons associated with it. We take this weird molecule here. It's called oxaloacetic acid. It's got a bunch of carbons, whatever. We add these together and shoot it through this whole mess. I always forget that it does that. I apologize. There it is. Okay. We're going to shoot it through this whole mess of reactions and whatever. What's the point of the Krebs cycle? That's what this is, by the way, the Krebs cycle. The whole point of taking that acetyl-CoA, combining it with the other thing, and putting it through the cycle is to kick off more carbon dioxide. Here's your carbon dioxide. Here's the carbon dioxide. There was carbon dioxide up here. And every time that you're taking off a carbon dioxide, as well as a lot of these other steps that are in the middle of all of this, you can collect electrons. So NADH, here's electron collection. Here's electron collection. Here's electron collection. This is FADH2. It's the first time we've seen that one, huh? And then electron collection, and then electron collection. Look at how many times we get to collect the electrons here. This is for 1 pyruvic acid going into 1 acetyl-CoA and 1 turn of the Krebs cycle. You get to do it twice for 1 glucose molecule, yeah? So that's the value of the Krebs cycle here. Collecting all of these electrons, breaking these bonds and reassorting them and whatever with these weird waste products. Luckily, during all of this, at some point, you get to take some crap and put a phosphate onto something and make it ATP. That's that substrate level phosphorylation we were talking about, but it's not even that important, okay? So I told you, glycolysis, the point, pyruvic acid. I'm just gonna put pyruvate, same difference, all right? All right, Krebs, the whole point are electron carriers, NADH and FADH2. And now I'm just putting parentheses, electron E minus, we'll just say that's electron, yeah? Electron carriers. That's the point of this. That's what I want you to take home. So remember also, if I were to ask you, in which cycle do we make NADH? We made some in glycolysis too. So we make it in both, okay? In which cycle do we make carbon dioxide? I'm not talking about the intermediate steps. I'm talking about the cycle. Is it glycolysis or is it Krebs? Krebs. We did not release a single carbon dioxide in the glycolysis. All we did, we would have had to shoot off a carbon, yeah? We kept our carbons. Can't make it there. That's not what's happening. So glycolysis, we're making pyruvate. That's the goal. Krebs, we're using that pyruvate essentially to make NADH and FADH. Those are our electron carriers. So I'm guessing that you probably can figure out next what's going to happen. We're going to use those electron carriers because what is that even for? This is the electron transport chain. We have redox carriers. They're just some molecules that are made to interact with electrons. That's all. Don't overthink it, okay? And essentially what's going to happen, let me just walk it through because I feel like it's easier to have like a drawing while it's happening. I understand better while it's happening as opposed to this, but you can look at it up there while I do this to understand what I'm drawing in case it's sloppy. Okay? So obviously bacteria have a thick peptidoglycan cell wall, they're positive. Yeah? That's its membrane. So that's its cytoplasmic membrane. All along the membrane, whether it's their membrane or the membrane on the folds of our cristae, in our mitochondria, doesn't matter. It all works the same way. All right. Here's our membrane. And on this membrane, we've got our redox carriers, or electron carriers, whatever it is. These protons that are going to interact. They're called cytochromes. Right? They're made specifically to interact with electrons. But these cytochromes, that make it clear that they don't have any weird whatever going on the inside. Cytochromes. Okay, we have our NADH. That's going to become NAD, whatever it's going to be reused, yeah, later, plus H plus. And you're like, wait. They're both positive though. Yeah. The electron's going here. Why it's really lonesome. You ain't connected to anything anymore. Okay? That electron's not connected to these molecules here. Right? We've got both NAD plus, H plus. All right, cool. We'll get to this now. Okay? Electron shovels on through. We're going to go through all of these molecules here. There's no other color. Go through all of these molecules here with our electron, right? Got a negatively charged electron and interaction with these cytochromes allows us to do one thing that allows that the most important thing that your cell will ever do in your entire life, right? You're going to take all this crap, all these other ions, protons, yeah, positively charged, whatever. And we're going to shuttle them outside. Like it shows in the picture, yeah? We're going to shuttle them outside because they're not in here anymore. They got shuttled out. Okay. So we have H plus. I'm just going to put pluses here so you know what is going on. I'm not going to work by all of it. This is the whole point of moving that electron through. We're creating this positive charge on the outside of the cells here. And what would that make this on the inside? Respectively, negatively charged, right? Let me tell you. When we learn about diffusion, things want to move from areas of high concentration to a low concentration. That's just kind of physics. So it wants to equal out on both sides. That's just physics. So what happens is, come through this wonderful, shiny, happy little channel. Okay? These guys will just move through this channel. We already know that positively charged protons can have an effect on a protein, right? Just because everything's a hydrogen bonds and how important is it, and blah, blah, blah. So we got all these positives going through this, causing changes in the shape of this protein, which this is a protein. And what that does is allows this protein to take ADP, make ATP. Y'all, I just told you how your cell is going to make energy without putting energy in. We're adding a bond without putting energy in. Okay? That is oxidative phosphorylation. Okay? That's what that is. When we mentioned it earlier, and I said how important it is to make your energy. That's what it is. Okay? We've used electrons, so it's oxidative phosphorylation. We just moved electrons through. Okay? Power it. Neat. So neat that we'll call this molecule up here, this protein. This is called ATP synthase. That's just what protein is called. It makes sense, appropriate for what's happening here. This is great. So now we've got a couple problems. Now we've got our ATP, and we can just make ATP and that nice and drive ourselves and whatever. But this relies on this gradient. You keep pumping in hydrogen ions through here, they're just still protons, even when they get back in. You're going to neutralize that difference. You cannot have it, right? You can't have that. So those N pluses, that's the problem, right? Here, we forgot about this, didn't we? What did I say about electrons? They don't just disappear into the ether. We've moved through everything. They need to go somewhere. It's build up there. What we do here, kind of pair it with this, take away that positive. We'll take a couple of those guys, actually. And then make water as a byproduct. That is where the electrons are going to go, as well as your built up protons. If you do not breathe in, you can't do this, okay? You won't be able to keep this whole system going. When you hold your breath, even right now, and you start feeling like you need to breathe, that's because you need ATP in your body. You need ATP for your heart. Yeah? That's how important this is. I mean, you probably never heard of this before, but it's unimaginably important in your body. Yeah? It is what does everything for you. You can't have your liver make bile or whatever for you without this. So, anyways, yeah, that's why you die if I suffocate you. Suffocating isn't just you don't have oxygen. Yeah, you don't have oxygen, but that oxygen, the reason you're hurting for it is that. The only reason you're hurting for it is that. Notice also that the oxygen that you just breathed in went and got used up to make water as a byproduct to take care of those electrons and those protons. Didn't touch the carbon dioxide. They are not related. You inhale oxygen, you exhale carbon dioxide because that's how it goes. But they are not, they didn't touch each other. A lot of people think they do. All right, we can see here in our little figure, we've got our oxygen over here for our aerobics, and then our anaerobes, not oxygen. That's all you need to know, okay? Not oxygen. Cool. This is what would happen in a eukaryote. You've got mitochondria to make all this energy for you. You're going to do this on the mitochondrial membrane called the cristae. Okay. We already talked about what these are and what they do. When you have the movement, I always forget to talk about this. When you move these protons across like this and build up this gradient because of that electron movement, yeah, okay, oxidative phosphorylation happens. But this part just right here, moving electrons and pumping across the protons, that's called chemiosmosis. Okay. Then, yeah, all right, proton motive voice. I always forget to talk about that. I get so excited about how it works. Yeah, so you have chemiosmosis, you move the protons out. That buildup is called the proton motive force. Kind of motive because that's what's motivating the whole process. They have names and you need to know what they're called. Right, we understand how that works. Okay, so this is kind of how the yield would go. And again, all I really want you to know, glycolysis, you're taking glucose. I'll be right there with you, I promise. Sorry, somebody commented in a chat. I promise that I will get it. I see this, so you can email me about it. So I'm assuming that you are talking about, why is this not letting me? I don't know what's happening anymore. Anyways, okay. Glucose to pyruvate. That's the point of that one. You already know it. Yeah. You make a little bit of this and some of this. You have to know that for the fermentation. Okay. This is that step where you turn that pyruvate into acetyl CoA. Okay. That's going to enter the Krebs cycle. We're going to move it through a whole bunch of molecules, kick out some carbon dioxide, accidentally make some ATP, but the whole point is making these electron carriers. This is the point for this, okay? Whereas glycolysis, this is the point. Okay, so we've gone from glucose to pyruvate and then made some electron carriers in the Krebs cycle. It was really the watered down version of it. And then shoved those electrons into the electron transport system, those negatively charged electrons, moving through those cytochromes. And then we will go ahead and cause that to make protons pump out to create the proton motive force. The proton motive force with those positive protons moving through the protein ATP synthase. You see how it has ACE at the end? What does that make it? Enzyme. Yes. So we're moving those protons through, causes it to do its job because of the protons being positive. We built up too many on the outside though, and then we got leftover electrons. So we use oxygen, half of an oxygen per water, to go ahead and take care of all that mess. Yeah, which is why we breathe in oxygen. So that is essentially how oxidative phosphorylation works and how we're going to get ATP at the end of that. So if all of this works like it is supposed to, in bacteria, you get 38 ATP. In us, eukaryotes, you get 36. That's just because you use some ATP to get crap going across the mitochondria. They don't have to do that as bacteria. Okay, they get a little more. I don't care about the water. I would be aware of the fact that you do not make carbon dioxide in glycolysis. Yeah, one way you can assure yourself of that. What's the point of glycolysis? Glucose, 6 carbons. Pyruvates, 2 of those, 3 carbons. Same carbons, you didn't lose a carbon, you didn't have carbon dioxide. Okay, cool. I don't need you to know every single little thing. Yeah, so anything except for oxygen that's going to make you anaerobic. All right, fermentation, like I said, 2 ATP is going to be made from that whole glycolysis cycle. So here it is, watered down again. You do make an electron carrier though. It is not worth it for a fermenter to make a proton motive force with that one thing. So it has to get rid of the electron associated with it and the proton somehow. Okay, that's its problem. After it makes its 2 minuscule little ATP, it has pyruvic acid to deal with and an electron to deal with and also possibly proton to deal with. We got to deal with these. Okay, so what do some bacteria do? They're going to kick off carbon dioxide and they're going to make some other crap and we're going to make ethyl alcohol. Here, we're getting rid of electron here. That's how it does it. That's nice. What's ethyl alcohol? It's drinking alcohol, ethanol. We also have another way of doing this. We can use that whole process, that pyruvic acid plus the electrons to make lactic acid. Guys, have you ever had kimchi or yogurt or something like that with the sour from the fermentation? There you go. Okay, so also these guys have to be complicated. I wouldn't maybe. Homolactic, homo, same. This is just lactic acid, right? It's not going to be anything exciting, okay? That's homolactic. Hetero is different. So we're going to have lactic acid, sometimes acetic acid, and you'll also have carbon dioxide, different stuff made here. That's really what it's telling you, okay? So not a whole bunch of different stuff, but a couple different things, right? Because next we have mixed acid fermentation. They're heterolactic. Okay, we're just talking mostly about lactic. Cool. Then we have mixed acid. Now we stepped away a little bit from that lactic. Now we're really mixing up our acids that we're making as a byproduct while we're getting rid of electrons and protons and whatever. You can make all of this at the same time. That's mixed acid fermentation. I don't need you to tell me which ones. Just be aware that it exists. Okay. We'll get into talking about where that's important whenever we get to that in the lab, but I don't really need you to worry about it here. I don't need you to worry about what this is called here, okay? But I do want you to know, we take a 6-carbon fatty acid. Now, I don't know if you guys know this, but fatty acids generally are not 6 carbons. They're like 20 carbons, right? Have you ever seen like unsaturated fats or saturated fats, things like that? That's what I'm talking about here, okay? They're usually long chains, but if you take a 6-carbon fatty acid, same as our glucose, and put it through the whole cycle, however you need to alter it to get it in there, but if you put it through there, you will get 50 ATP. 6 carbons still, you're gonna get 50 ATP. Whereas we get 38 from a 6-carbon sugar, glucose, which is our bread and butter explanation for this. Now, I don't need you to know too much about the details, but I want you to be aware of this. 50 ATP is a lot more energy. This is why your body stores energy as fat. It's more energy efficient. That makes sense, right? A lot more ATP. You can also get some energy out of like proteins and breaking them down and amino acids and whatever. You often get a little bit less energy from this. That's where, you know, keto diet comes in because you're getting less. So your body's doing more work for less. And that works in your favor if you're trying to burn calories and stuff like that. Yeah, that's the idea. Okay, amphibolism is just a term that refers to the fact that we have all these pathways working together. You break some bonds here, you can use the byproducts over there to make these, and then whatever. You don't really need me to explain that, right? Like we all know that breaking something down, you can use the parts from that to build another thing. We're gonna shuttle it through the frickin' aerobic whatever. Anytime you're building cells, you need new membranes. You need to double everything that you have going on everything, your ribosomes, your everything. It doesn't even matter. All of it needs to double. You need to go through anabolism, which is building things. Yeah. So you need energy for that, energy in the form of ATP. So think about it that way. You also need the byproducts from all of this to build it all. Whew, we're going to just make it. Okay. All right. Light dependent reactions. Just like it sounds, you need light for this part to happen. All right. So that's over here on the left side. The light dependent reactions in photosynthesis. We're going to have sunlight hitting you. We have some little molecules, the pigments like chlorophyll. They get excited and release some electrons whenever they're hit by the photons. Okay. Like these proteins that were over here, basically like instead of being donated the electrons, we're going to be using light literally to release electrons from the pigments and send them through. We're going to make ATP that way in a plant. Okay, great. We make ATP. Isn't that exciting? Yeah, I think so too. So we make ATP. It's not very much. Okay, it's like 2 ATP, I think, from one of those. What do we use that ATP for? The light independent. It doesn't need light is all it means. It doesn't mean it has to be in the dark. It just doesn't need light. We call it the Calvin cycle. Okay. In the Calvin cycle, I don't like this slide. In the Calvin cycle, we're going to use that ATP and some carbon dioxide and put them together to make our good old friend glucose. And guess what plants do with it then? Aerobic respiration. They have mitochondria too, y'all. They do the same things that we do. I think that is so cool. You take inorganic carbon dioxide and some light and get enough energy to make glucose and then kick out 36 ATP. That is crazy cool. Plants, man, they're cooler than us. That's all I really need you to know about that. But this is what it looks like if you want to think about how it works. I don't need you to know the details other than the fact that, yes, they're going to be using light to make ATP, but that ATP is used to make glucose, and the glucose is just the same thing that we do with it to make ATP. So they do need oxygen. If you take a plant and put it like, okay, so we have photosynthesis that makes oxygen, but it doesn't really make enough necessarily for a plant to sustain itself unless there are certain other factors going on there. So most plants, if you just take a basic plant and put it like under something that's completely airtight, most of the time it will suffocate. That's real. It needs oxygen too. You can't make all of its oxygen. So this is the chemical stuff involved that you don't need to know. Like I said, we're putting in carbon dioxide, using our ATP that we made from the light, and then we're getting out glucose in something called the Calvin cycle. Okay. Okay, we have different kinds of photosynthesis. They either make oxygen or they don't. Oxygenic makes oxygen, and oxygenic does not make oxygen. Don't really care about the details beyond that, okay? Obviously, oxygenic, that makes this plants, algae, the green bacteria, all that. All right, so we just talked about this. So you probably don't need me to answer it or anything, but why do plants need to synthesize glucose?